Welcome to our fifth webinar, sponsored by the donors and friends of the Brain and Behavior Research Foundation. To those of you who are joining us for the first time in this series, thank you very much, and to those of you who are returning, welcome back. In fulfilling the mission to the 1 in 4 people diagnosed with mental illness, the Brain and Behavior Research Foundation's Meet the Scientist webinar program is yet another example of how we realize our commitment to educating and informing our audience. As always, these webinars are free of charge and are available to the larger community on our website. And despite economic uncertainty and ever-decreasing federal funds for basic research, the Brain and Behavior Research Foundation continues, as it has for 24 years, under the name NARSAD, to advance the field of mental health through mental illness research and education. By providing much-needed catalytic funding for promising research ideas like those you'll hear today, and through our ongoing symposia and publications, our commitment to you remains steadfast. To all of you who support our efforts, thank you. We sincerely appreciate your key role in our joint success and hope that we can continue to count on you with your thoughtful contributions. I am now pleased to introduce you to a very good friend of the Foundation, Scientific Council member Dr. John Krystal, as today's presenter. Some of you may already know Dr. Krystal from our 2010 Palm Beach Symposium. Dr. Krystal was also featured in our most recent Winter Quarterly magazine, which is available on our website, addressing anxiety disorders. Dr. Krystal is the Robert L. McNeil Jr. Professor of Psychiatry at Yale University and chairs the Department of Psychiatry. He is also Chief of Psychiatry at Yale New Haven Hospital. Today's discussion, Lessons Learned from Experimental Psychopharmacology, will cover, among other things, the effects of drugs on behavior. Please welcome Dr. Krystal, who will speak for approximately 30 minutes and then answer some questions as you pose them from our audience. Thank you. Dr. Krystal?
Thank you. Thank you, Benita. And let me just say it's a real pleasure to speak as part of the Brain and Behavior Research Foundation. This is one of the world's wonderful organizations whose mission is so important to the work that we do at Yale and at all the departments of psychiatry around the country. Today I'm going to be talking about work that we've been doing for over 20 years to develop new approaches to the treatment of schizophrenia and depression. And in this work, we, by necessity, collaborate with a variety of pharmaceutical and biotechnology companies that I list here. The question that has driven our research is whether neuroscience or translational neuroscience can lead us to new treatments for illnesses like schizophrenia and depression. These are common, distressing, disabling, and even lethal disorders, despite often the best treatments that we have to offer. Both disorders are commonly treated with antipsychotics, and with perhaps the exception of clozapine and electroconvulsive therapy, most of the medications that are used to treat basically have similar efficacy profiles, and we've had these medications for now 50 years. So there's growing industry concern about investing in psychiatry drug development with a number of recent failures in the development process. That bad news is balanced by really important good news, that we have better animal models, better insights into the genetics of these disorders, better insights into the brain networks underlying the symptoms, and a variety of novel intracellular targets. With this explosion in knowledge, we're now faced at times with the question about how do we choose from among the many new potential targets to test in the development process. I'm going to be talking about schizophrenia and depression today. I'm going to begin by talking about how my laboratory and collaborators have been probing the glutamate system of the brain in schizophrenia using a drug that blocks one of the glutamate receptors. That drug is called ketamine. Now, we got onto this work in the late 1980s, beginning with our experience with clozapine, feeling that while it was an advance, it wasn't really going to solve all the problems of schizophrenia. And so in our work, we tried to shift the focus of what we were trying to do from the dopamine system, which begins in a very primitive part of the brain and modulates the higher centers, to the intrinsic biology of the cerebral cortex, in part because we viewed disorders like schizophrenia as disorders of the development and function of the cerebral cortex. Now, our work focused, instead of the dopamine system, it began with a focus on the glutamate system. And glutamate is the main excitatory neurotransmitter of the brain. Particularly, it's responsible for all excitatory cortical output. And there are three main types of glutamate receptors, one of which I'm going to really focus on today, which is called the NMDA glutamate receptor. But there are two other types that I'll touch on very briefly. One is called the AMPA, and the other family are called metabotropic glutamate receptors. It's important to remember that when we were beginning this work, one of the challenges of the field was to really understand the whole cluster of the symptoms of schizophrenia. That most of the work had previously focused on the positive symptoms like delusions and hallucinations, and yet we were aware that negative symptoms such as blunted emotion and withdrawal, or thought disorganization, or cognitive impairments were very important to how people were able to function when they had schizophrenia. So in our work with ketamine, one of the things that we were struck by right from our initial study was that ketamine was a pharmacologic probe that would be able to very briefly and in ways that could be studied safely produce symptoms associated with both the positive and the negative and the disorganization and the cognitive symptoms of schizophrenia. And this was really the first kind of demonstration that a single drug, a single mechanism could produce this entire profile. And the slide that you're looking at right now, it was a study that illustrated that point. What you see in the black and the red lines are the effects of ketamine with and without amphetamine. And what you see with the green line is the effects of amphetamine by itself. So what you see on the left half of the graph is the fact that both amphetamine and ketamine are able to produce symptoms in healthy subjects that resemble the positive symptoms of schizophrenia. But if you look on the right-hand side of the graph, what you see is that amphetamine does not produce any of the negative symptoms that are produced by ketamine. And so that ketamine was really a different kind of model that had the possibility of providing insights to the broad spectrum of symptoms of schizophrenia and not just psychosis. Now, why would NMDA receptor function? Why would be a drug that blocks NMDA receptors give us these kinds of insight to schizophrenia? There are many ways of answering that question. I'm just going to show you a few pieces of data from our work. In this slide, you see some work that was conducted in collaboration with Lynn Pawlowski, the late Lynn Pawlowski at the Institute of Psychiatry in London, where we used a radio tracer for the NMDA glutamate receptor in the context of SPECT neuroreceptor imaging to show for the first time that there were changes in patients in vivo, in fact, reductions in the levels of the NMDA receptors in the brains of people with schizophrenia. But what about functions associated with the NMDA receptor? We collaborated with our longstanding collaborator, Patricia Goldman-Rakic, obviously, who passed away not too long ago, to study the role of NMDA receptors in working memory, in human working memory. And we adopted a task from her studies of the non-human primate where stimuli were presented in various locations and people had to encode that information into working memory. Then there was a pause when people had to maintain that information in working memory, and then there was a response phase where people had to determine whether stimulus had been previously presented in the place of the probe. And all while people were performing this task, we were measuring their brain activity with functional magnetic resonance imaging. What our group found was that in the lateral prefrontal cortex, an area of the brain involved in working memory, as people were encoding the information into working memory, healthy people, this is the top line, you see that there was activation of the lateral prefrontal cortex, which was sustained while they were maintaining the information online. And there was another activation of this area during the response phase. People with schizophrenia were activated this region as well, but there was a lower level of activation during the maintenance phase of working memory, and also a lower activation in the response phase of working memory. This is consistent with the idea that reductions in prefrontal cortical function are contributing to working memory deficits associated with schizophrenia. So one of the things we wanted to know was, when you give a drug that reduces NMDA receptor function, do you see a similar change in the pattern of brain activity? And this is exactly what we're finding. Here you see the same person, or the same group of people, getting on one day saline and activating their prefrontal cortex when they're encoding working memory, keeping it activated when they're keeping the information online, and activating it again during the response phase. And you see that ketamine attenuates this activation of the prefrontal cortex. So what I've shown you so far is evidence that that ketamine, a drug that blocks this particular glutamate receptor subtype, produces changes in brain circuit function that mimic the dysfunction of those same brain circuits in schizophrenia. So this yields one novel idea about the treatment of schizophrenia, which is, if one could enhance NMDA receptor function, maybe one could normalize the disturbances in these brain circuits and reduce symptoms of schizophrenia. The most common strategy for enhancing that has focused on a particular substance, two substances really, glycine and D-serine, both of which are capable of partnering with glutamate to activate NMDA glutamate receptors. Now, NMDA glutamate receptors are channels or pores that allow calcium to enter the cell. So when glutamate binds and either glycine or D-serine binds to the NMDA receptor, then calcium can activate the cell via this channel. Now, we have conducted some preliminary data using very high levels of intravenous glycine using the same working memory paradigm during functional magnetic resonance imaging. And what you can see on the left side is that if a person is healthy and they get glycine, by itself it has no beneficial effect on the prefrontal cortex. However, if there's a deficit in the function of the NMDA receptor, this is the right side of the graph, a deficit in the NMDA receptor function produced by ketamine, that's the blue line on the right side of the graph, then, and only under those conditions when you give glycine, do you see what looks like a normalization of the function of the prefrontal cortex. Now, glycine is not a very workable treatment, and although people have tried to give very high doses of glycine as a treatment for schizophrenia, the results have been kind of mixed. Instead, what the pharmaceutical companies have focused on is a novel way of raising synaptic glycine levels that has to do with the glycine-1 transporter. These are the Hoovers. If we could block the activity of those transporters or vacuums and allow more glycine to be in the synapse, one could raise glycine levels and enhance the stimulation of the NMDA glutamate receptor. What you see here in this slide is the results of an initial study conducted by my colleague, Dr. Cyril D'Souza at Yale, that shows if you pre-treat someone with a glycine-1 transporter inhibitor, then one can alter, essentially reduce, the behavioral effects of ketamine in healthy people. These kind of data lend support to the idea that in patients with schizophrenia, one might see a benefit by raising synaptic glycine with a glycine-1 transporter blocker or a GlyT1 inhibitor. Now, what's interesting is that in November of 2009, Roche issued their first report of a preliminary study suggesting that when a GlyT1 inhibitor was added to ongoing medication treatment for schizophrenia, that they had both a reduction in negative symptoms and improvement in social functioning. So these kind of data, which emerge from a translational neuroscience process, suggest that circuit effects of NMDA receptor antagonists resemble some aspects of schizophrenia, and that promoting NMDA receptor function may attenuate some circuit deficits and may hold the promise to reduce possibly negative symptoms and other symptoms associated with schizophrenia. I'm going to now shift to a second consequence of the ketamine model, which has to do with ketamine mimicking the effects of deficits in GABA or inhibitory nerve cells in the brain. And the issue is that by disinhibiting brain glutamate systems, ketamine may amplify some of its disruptive effects on brain function. And this suggested that drugs that reduce glutamate hyperactivity might treat some symptoms of schizophrenia. This is just a figure that grows out of the work of David Lewis and summarizes the work from a number of groups that suggest that GABA or inhibitory nerve cells in the cerebral cortex may be dysfunctional, so that in addition to glutamate synaptic dysfunction, we may also have GABA or inhibitory synaptic dysfunction as well. One of the questions in the field had been, how do we know that these inhibitory deficits have meaningful functional implications for patients with schizophrenia? In order to study that question from a pharmacologic point of view, we began to study in people the effects of a drug called iomazenol, which causes a slight deficit in GABA function. So it's the opposite of Valium that enhances GABA function. Here is a drug that causes a transient and slight deficit in GABA function. And the question is, if you create a deficit in GABA function, could that contribute to the hyperactive response to amphetamine associated with schizophrenia? If that were the case, amphetamine, which normally doesn't produce psychosis, would produce psychosis briefly following pretreatment with iomazenol. And this is exactly what our preliminary data suggest, which is that iomazenol by itself doesn't really do much. Amphetamine administration by itself doesn't do much. But if you cause a GABA deficit first with iomazenol and then give amphetamine, then you induce a transient psychotic state, which suggests that the GABA deficits in schizophrenia do have the possibility of being relevant to the symptoms and cognitive deficits of that disorder. So how does that link in with the with the NMDA receptor story? It turns out that when you give ketamine, you prevent or reduce the activation of certain GABA nerve cells in the brain, which reduces the inhibition of other glutamate nerve cells in the brain, resulting in increased levels of the release of glutamate. And that glutamate is functioning in a pathological way because of the blockade of the NMDA glutamate receptors. So we can mimic some aspects of GABA neuronal dysfunction by giving a drug like ketamine, which suggests one possible novel way of thinking about how to enhance the treatment of schizophrenia. If glutamate systems are hyperactivity, then one might be able to, by toning down the glutamate system, improve the functionality of glutamate neurotransmission in the brain. And the first test of that hypothesis, we used an anticonvulsant drug called lamotrigine, which does reduce glutamate in certain models. And what you see on the left-hand side is the ketamine and ketamine plus lamotrigine test days. You see that in blue, lamotrigine, when pretreated with lamotrigine, you have a reduced positive symptoms produced by ketamine. And on the right side, you see that in blue, again, when you receive lamotrigine, you have reduced negative symptoms produced by ketamine. So lamotrigine was the first agent that we found to attenuate ketamine effects in people. And other groups built on these data to conduct clinical trials in patients who were very treatment-resistant. And in these very treatment-resistant populations, but it turns out potentially not in all populations, there was some benefit of adding lamotrigine on top of antipsychotic medications, particularly clozapine, to enhance the effectiveness of the treatment of schizophrenia. However, another medication has emerged as an even more exciting way to approach this problem, taking advantage of the normal mechanism of the brain for feedback inhibition of glutamate nerve cells. One of these mechanisms is a glutamate receptor called an mGluR2 receptor, or a metabotropic glutamate receptor type 2, which provides feedback inhibition on glutamate release. Beta-mogadam in animals showed that if you used a drug that stimulates this receptor, you can attenuate PCP effects and glutamate hyperactivity in animals. Based on that work, we tested this medication, LY354740. Again, it's a mGluR2 stimulating medication. And we showed that it reduced the disruption in working memory in healthy people. On the black, you see the effects of the drug by itself. In red, you see that under the placebo condition, ketamine is impairing working memory, and that impairment is attenuated by giving more and more higher doses of this drug. Based on this, Lilly conducted a study of a drug related to that prior drug called LY2140023, which showed it was about as effective as olanzapine in the treatment of schizophrenia. The exciting thing about this slide, if you think about it, is that in 50 years of the treatment of schizophrenia, since the introduction of clozapine, every antipsychotic worked by reducing the stimulation of dopamine D2 receptors. These data, building on a translational research model, was the first time we had a medication which emerged from a rational translational neuroscience process, which worked by a novel mechanism, in other words, didn't block the dopamine D2 receptor, and yet seemed to be effective for the treatment of schizophrenia. Now that was pretty exciting. The news in subsequent studies with this drug has been more mixed, and we're still waiting to see whether something useful will come out of this approach. But from a conceptual point of view, and in terms of a new generation of novel treatment mechanisms, I think was a very exciting finding. So in summary, what I've shown you so far, and we're about done with the schizophrenia part of the story, is that drugs that enhance NMDA receptor function or reduce deficits in cortical inhibition are novel treatment strategies for schizophrenia that emerge from a translational neuroscience, experimental, human psychopharmacology approach. I thought I would just, in a way, give you a teaser to show another way in which we're using ketamine to study the brain, which is as a antidepressant medication. You might, and I'm going to just talk briefly about glutamate alterations associated with depression and the possibility that it might lead to glutamate-based treatments for depression. Now, early on, we had found some evidence, and other groups have found evidence in magnetic resonance spectroscopy studies of elevated levels of cortical glutamate in depression. And we believe, based on a variety of different kinds of data, that these increases in glutamate levels may reflect, in part, a reduction in the function or even reduction in the number of glial or supporting cells of the brain. These glial cells are responsible for taking up and metabolizing glutamate so that there's not overstimulation. So in depression, where we believe, based on postmortem research studies and other data, that there are glial deficits, the predicted consequences of those glial deficits include excessive stimulation of the synaptic glutamate receptors, which can produce neurotoxicity or cause loss of the function of those receptors. And in addition, excessive stimulation of two categories of receptors. On the neuron on the left, which is sometimes called the glutamate-releasing or presynaptic neuron, you can get excessive stimulation of that same mGluR2 receptor, but in this case it's causing these neurons to fire insufficiently. Or on the right-hand side, on the green neuron, which is called the postsynaptic neuron, you see that there are some glutamate receptors that are not in the synaptic space between the nerve cells. These are sometimes called extrasynaptic glutamate receptors. And when they are overstimulated, it causes atrophy of the synaptic connections and the loss of a structure called a dendritic spine. So when we try to move from pathology to treatment, you might ask the question, glial deficits cause problems. How can they be fixed by new glutamatergic treatments? One problem being a suppression of glutamate release. Now, I had just shown you some evidence that NMDA receptor antagonists inhibit GABA neurons and cause the release of glutamate. So NMDA receptor antagonists are one strategy to achieve this. Another problem that I just mentioned was that you might get overstimulation of these extrasynaptic, not in the synapse, but outside of the synapse, NMDA receptors. And NMDA receptor blockers would also be a strategy for producing that effect. So based on this kind of rationale and other kinds of thinking, we tested NMDA receptor antagonists in patients with depression. And what we found was really quite remarkable, that the drug did produce the expected transient euphoric effects and transient cognitive and perceptual changes that we saw in other populations. But if you look at the top row of the graph, what was really striking in this group of treatment-resistant depression patients was that their level of depression was, as a group, was down by about 6 hours, and by about 24 hours, many of the patients in this group were clinically remitted and not higher euphoric, but just back to their usual selves. And this lasted, even though this drug only has effects for about a half hour or an hour or two, these antidepressant effects emerged within hours and stayed for days and sometimes weeks. Our findings were replicated by several sites now, particularly Carlos Serrati and Husseini Manji when they were at the National Institute of Mental Health Intramural Research Program. And this research has now stimulated basic scientists to help us to understand the cellular and molecular mechanisms underlying this rapid antidepressant response. As I mentioned, one part of the story is what I sometimes call the GO pathway, which is by disinhibiting glutamate release, we activate the AMPA receptor. There is a variety of signaling molecules that are involved, particularly the AKT-mTOR pathway, and it causes the growth of new dendritic spines, the structures that enable connections to be made in the brain, which disappear under conditions of severe stress or in depression. Ketamine also affects another pathway you might call the STOP pathway, which acts via elongation to factor 2 to reduce BDNF levels and to suppress. So it enhances the GO and it reduces the STOP, and we think that these two pathways help us to understand how you get such rapid and profound antidepressant responses. It also suggests that there are certain other targets besides the traditional receptors that one might focus on in new treatments for depression. So in summary, glial deficits may contribute to glutamate synaptic dysfunction in schizophrenia, and novel pharmacologic strategies like NMDA receptor antagonists may point us to new treatments. So overall, what I've shown you today are examples from our work in schizophrenia and in depression that have where understanding the underlying neurobiology and carrying that over into experimental psychopharmacology have pointed us in the direction of new treatments. For schizophrenia, this is in the area of enhancing NMDA glutamate receptor function and reducing glutamate hyperactivity. And in the case of depression, it's involved in compensating for the impact of loss of glial function, both with regards to the enhancement of AMPA receptor function and the block of the extrasynaptic NMDA receptors. Let me acknowledge, of course, my collaborators in this work and supporters in this work, particularly the Brain and Behavior Research Foundation, which have helped us at critical points in developing this story, and my collaborators in the depression work as well. So thank you for hearing this story out, and I'd be very happy to take any questions or hear any comments that people would like to make about this work.
Well, thank you, Dr. Krystal. We do have a couple of questions that have come in, and of course I'm still welcoming others, but this seems to be a theme that we hear from many of our constituents when we hear about pharmacological advances. It's the when, it's the how quickly, it's the studies are ongoing continuously, but when can we anticipate that some of these activities will result in clinical trials? And if there are current clinical trials related to any of the work that you're working on, in what phase, perhaps?
Sure. With ketamine, the story is really quite interesting in that it's an FDA-approved medication. So we're not in the situation of going through all the development process to have FDA approval, but rather in the case of the antidepressant effects of ketamine, we're at the stage where we're trying to best understand how to utilize and how to maximize its benefits. So I am aware that there are a number of physicians around the country who have started to employ ketamine in their practices, but I'm not really sure that we have all the information that we need to know how to best advise doctors to use this medication. Clearly, it can act very rapidly. In some people, we know that the effects can be sustained for 3 weeks or more, but to me, it's still a research-based approach, and I'd be happy if people have questions about where those studies are that are studying ketamine and related drugs to try to help people out. They can contact me directly at john.krystal, Krystal, @yale.edu. With regard to the other treatment approaches, the glycine-1 inhibitors, these are drugs, all of which are being developed by pharmaceutical companies, and where for example, I believe that Roche has a very active program around the world where they are conducting studies with their glycine-1 inhibitor. And I would suggest contacting Dr. Daniel Umbricht, Umbricht, at Roche to find out whether or not there is a clinical trial close to your home to see if for participation. There are some other companies that are also doing studies. They're not all at the same stage of research. Roche's drug is the farthest advanced in the development process. With regard to lamotrigine, that's a drug that every psychiatrist can prescribe and is available. And with regard to the mGluR 2/3 approaches, the drug that's farthest along is the Lilly drug, and they have ongoing clinical trials with that drug as well. And the person to contact to find out whether or not there's a site of that, where that drug is being tested close to you, is Dr. Bruce Kynon, Kinon, at Eli Lilly and Company in Indianapolis, Indiana. So that's the buzz on those drugs. There are other drugs that build on these models that are in development and being tested by other groups, and I'd be happy to do my best to try to connect you to those if you're interested.
Well, I have to tell you, Dr. Krystal, offering your email and additional consultation on some of these therapies that you've shared, or the drugs that you've shared, is very, very generous. And so thank you very much for taking that extra step with our participants, as well as the other information that you, very specific, just really very good. Thank you. Mm-hmm. Here is another question from one of our current participants. Would this new approach, and this is in relationship to depression, would this new approach avoid the bad reaction to current antidepressants in bipolar depression?
So it's an interesting question. So the problem that we sometimes get into with bipolar depression is that that antidepressants in some people can increase the risk of cycling from depression into mania or to other kinds of mixed affective states. And there is one study with ketamine in patients with bipolar II depression that came out of the National Institute of Mental Health, which suggested that ketamine was as effective and with a similar profile of efficacy in bipolar patients, bipolar depressed, as in unipolar. There is one case report of somebody with bipolar disorder, I think bipolar 1 disorder, in other words, with manic and depressed phases of their illness, where they had some increased cycling after a ketamine injection. So what that says is that while overall it's probably reasonably well tolerated, given the amount of information we have, which is pretty limited, we have to be obviously very cautious about drawing any firm conclusions.
Okay. And I guess in relationship to that, I'm not quite sure that I understand this question, but could you comment on a few, and it says S/E, major, of inducing NMDA activity for treating schizophrenia? Do you follow that?
Could you, what's the beginning?
Could you comment on a few major side effects? That's what he meant. Side effects.
Okay.
Okay. Thank you.
Yeah. So these are very interesting drugs, the glycine 1 transporter inhibitors, and in fact glycine and D-serine. So glycine is a constituent of the food that we eat, and we have high levels of it in our body. And as far as I can tell, raising the level of glycine has no, at least over the short periods of time that it's been studied, have no real effects of consequence. D-serine is an unnatural amino acid. In other words, it's made in very small quantities in very specific cells in the body. It doesn't circulate in high levels in the body, so that when you take high levels of it, there are parts of the body that are exposed to unnaturally high levels of D-serine. Overall, it seems to be quite safe. Based on some studies in dogs, but not other species, we're watching the effects of D-serine carefully, because in dogs there are some adverse effects on the kidney. So far in humans there's not been any sign of adverse effect. The GlyT1 inhibitors, as far as I can tell, are similarly relatively safe. However, there's one GlyT1 inhibitor that's being tested in Taiwan, which is called sarcosine, and there are some concerns, safety concerns with sarcosine that have prevented it from being studied in the United States so far. So overall, these are very safe drugs with very few side effects, a little bit of sedation with the GlyT1 inhibitors, so that's very encouraging.
A question just came in. What about indications for pregnant women? Are they considered safe?
Oh, we haven't gotten there yet with any of these drugs. I don't believe any of the medications have yet been tested in pregnant women. The only medication that we know of is lamotrigine, and lamotrigine is, as far as I can tell, no worse than other anticonvulsant medications, but as a class, the anticonvulsant medications are a class where there are some concerns about effects on the developing fetus. So I think it's too early to tell with the other medications.
Okay. You may have addressed this, but let me just make sure so that the participant's direct question is asked. How does ketamine affect the positive symptoms of schizophrenia, if at all?
Yes, interesting question. So in the doses that we've been administering yet, there are some groups that have studied ketamine effects. So Dr. Cal Taminga and Adrian Lottie, when they were at the University of Maryland, and Dr. Alan Breyer and Anil Malhotra when they were at the intramural program at the National Institute of Mental Health. Both of these groups studied ketamine effects in patients, and what they found was basically that there was a transient worsening of the typical symptoms of their illness after ketamine was administered. There's not that much data, but there's a sense from these investigators that perhaps the effect is a little bit more pronounced in patients than in healthy subjects.
Okay. And we do have one last question, I believe, unless there are others coming in. Is there any evidence that the new glycine or glutamate agents may contribute to metabolic syndrome, as we have seen with the atypical antipsychotics?
That's an excellent question. So glycine, when you think glycine safety, you should think tofu, because tofu has very high levels of glycine. And so consuming that doesn't seem to have any obvious metabolic effects. The mGluR2 agonists have as a side effect in some people nausea. And metabolically, if anything, it's associated with weight loss rather than weight gain. And this is a very interesting it creates a very interesting kind of synergy with antipsychotic medications. So antipsychotic medications are anti-nausea medications that produce weight gain. And mGluR2 agonists are medications that can cause nausea but cause weight loss. So if you combine these two medications, maybe you won't have any nausea and maybe you won't have any weight gain. So that it's conceivable not only would you have synergy with regard to the control of the symptoms of schizophrenia, but this might also be a strategy for managing some of the side effects of the antipsychotic medications.
Okay, and I think your continued answering the questions is stimulating more questions. So there are just a few more if you have time.
Sure.
What is your opinion of oral ketamine treatment for depression?
Yeah. So there are two formulations of ketamine that are I believe will be studied over the next few years, intranasal and oral. And I think my prediction is if they can get the doses high enough with these delivery routes that it is likely that you will see similar efficacy, which would be nice because the way ketamine has been studied up until now is entirely been involving intravenous ketamine administration. But using an oral or intranasal formulation would be a path to wider implementation of this approach if it turns out to work out well. But there's nothing published yet with it. I think there may have been maybe a case report about oral ketamine, but I haven't seen any studies.
Okay, and there's one last one that's very personal. Well, not very personal, but from my brother's anecdotal experience, smoking really relieves his schizophrenia symptoms, and when he quits, they get much worse. Mm-hmm. Are there any current experiments with nicotine related to this phenomenon?
Yes. And so we're studying nicotine and nicotine-related agents both in the context of the ketamine models where the findings haven't been as robust as in some studies of patients with schizophrenia. There are many different kinds of nicotine receptor subtypes. It's not entirely clear yet what the best approach will be, but I think this is an area that's being studied very intensively, particularly for a receptor called the alpha-7 subtype of nicotine receptor, drugs acting at that receptor. The one thing that we've learned from nicotine, though, about smoking is that the effects of smoking can be misleading, because one of the things that sometimes happens is that what people often describe in heavy smokers, what people often describe as the benefit of smoking, turns out to be, with careful study, the suppression of the negative effects of withdrawal from smoking. And so we're very clear that you can suppress the withdrawal-related worsening of symptoms and dysphoric mood and cognitive impairments. Whether you can really achieve true benefit in non-smoking patients, that remains to be seen yet, but is an important question.
Well, and there is one last question that, and I think we then wrap it up, but how often would the intravenous ketamine need to be taken for schizophrenia?
That's an excellent question. I think we've been working with the idea that a model for ketamine treatment might be something similar to electroconvulsive therapy. So it's almost like electroconvulsive therapy without the convulsive therapy. In other words, so we think that there'd be a period of time that maybe 2 or 3 times a week you would get a treatment, but then over several weeks that those episodes would be spaced out farther and farther, and that only maintenance intervention would be needed at that point. This is a theoretical model based on some of the similarities in the biochemical changes produced by ketamine and electroconvulsive therapy, but is supported by data at least 3 weeks out, where they have given ketamine 3 times a week and sustained remission of depression for that time. So it's an encouraging story, but I think one of the important issues raised by this is that it almost suggests a really important new idea, that there may be some treatments that work to induce remission, in other words, to make the symptoms go away, but it's possible that these medications may not be the best strategies for maintaining remission once it occurs. So in the case of medications from each of those classes, ketamine is perhaps our ideal or prototype remission-inducing agent. We don't have any treatment that works any more quickly than that, within 6 hours. But we also know that there are people who don't have an antidepressant response to lithium, but we know that lithium for some people can prevent relapse or worsening subsequent return of depression. And so that might be a prototype for a kind of a treatment that's involved in preventing or maintaining benefit or preventing relapse. And so we need to think about medication treatment for depression in a more fine-grained way than we have in the past, because we are starting to have medications which are shedding new light into the process of treating depression.
Dr. Krystal, you're just absolutely wonderful, and our participants have said so as well in their comments. And I'd like to thank our participants, and all of the questions have been wonderful, no matter whether I understood them or not. That's pretty irrelevant. You did, and thank you for helping me through that one in particular. Mm-hmm. Your research and your leadership and your support of the Brain and Behavior Research Foundation has been phenomenal, and we appreciate everyone who's joined us on today's seminar, and we hope you'll consider continuing to extend your support to enable us to fund more research. And I wanted to share one last thing to those of you who are still on the phone. Those of you in the metro New York area, please be reminded that on Wednesday, October 26th, you're invited to our 23rd Annual New York City Mental Health Research Symposium. You can get all the information online, or, of course, call us, and we'd be glad to share additional information personally. Thank you, everyone. Thank you, Dr. Krystal, especially, for participating. And we have one last webinar in this series on October 5th with Dr. William Bunny. So we look forward to your participation then again. Thank you.
Thank you.